approach that we used for analysis of COVID data that came our way, and I'll talk about that briefly. And I know that AI is some sort of a overhyped terminology these days, but I think Anton kind of laid the foundation for my talk really well. Thanks, Anton, for that. I think the fact that there has to be the functional correlates to any kind of model that you build. Otherwise, you end up looking at diagnostics and not potential therapeutic targets is very, very significant. So I'm going to be focusing on Berg's approach to mapping targets in disease using a different approach that is not functional genomics based, but it's a mapping of the causality of various molecules in a system. And then I will walk you through one example that we were able to pick a target for oncology, validate that, actually develop some molecules. And we are actually right now looking at IND enabling studies and taking it to IND next year. So that's just a disclaimer from Berg. Just quickly going through the essence of the platform or the technology that we use. I mean, the traditional approaches have always taken some sort of a unidirectional approach towards doing discovery, preclinical, clinical. But what we do at Berg is to put the patient and patient samples up front. As you can see on the right here, we digitize patients' information in terms of just not the clinical outcomes or any kind of molecular outcomes, but even going beyond genomics data that we possibly can. So we put the patient front and center of our discovery process, and for that reason, we spent the first many years since the company's inception in inking some of the deals with big hospital centers and foundations and academic centers that actually had access to some of these patient samples. So we focus mostly on oncology and neurology for now. And so we kind of went in that direction of seeking partnerships with, the Parkinson's Institute for Parkinson's disease and DOD for breast cancer tissues and samples and so on and so forth. So what my point here is that we put the patient samples up front and take them through omics technologies, omics analysis, identify targets, and then go through the process of discovery and preclinical validation, as well as drug discovery and development. So in a sense, what we do for target ID, what we do is trying to map causation within a system, a disease system. Causation as in, what drives what, other than a correlation. So from that, what we get is directionality between two molecules. What is driving what? It could also be what is repressing what, or what is kind of agonizing what. So we get both those kind of information using this approach. But prior to that, what we do is we take these samples, the first column on the left, we take these samples from various patients, whether it is Buffy coat, plasma serum, saliva, cerebrospinal fluid. If there are cells that we can obtain, we carry out cellular perturbation experiments, kind of perturbing disease, mimicking disease systems. And then from that, we move on to data acquisition, looking at metabolomics, lipidomics, proteomics, and genomics, profiling these samples, whether it is serum plasma or whether it is cellular samples, and then creating that causal network. So this network here, one thing I need to clarify is it is a de novo network. It is not based on any inputs from literature or something that is already known or published. This is a purely data-driven de novo approach of building a string of networks into some sort of a huge hairpin ball kind of a massive network, as you can see here. So in the process, we like to call this a white box approach, because all the algorithms, we are able to kind of look at the algorithms at work, and we are able to kind of QC them in real time. So the whole Bayesian, this is based on Bayesian, the causal mapping. I should take a step back and say that the causal mapping is based on a Bayesian algorithm, which is based on inferential causality. And so out of that effort, what we get are these networks, de novo networks. And from there, we are able to slice those networks depending on the questions that need to be answered. For example, what is the key driver of, in the case of COVID-19, one of the things we did was, what are the proteins that are directly connected to viral titer, for example. So in oncology, that could be, what are the proteins that are directly connected to proliferation or apoptosis or EMT or cellular migration, things like that. So based on the design of the experiment and the clinical question, we are able to create these data slices, what we call as delta networks, and simplify those networks into slices like what I have in, so basically that involves some topology analysis, as well as some additional analysis that we are able to perform, leading to this slice of data. And from that slice of data, we're able to create a list of various targets, what we call as a rank ordered list, that now allows us to kind of, go back and figure out what are the best targets to work with, and or what are the best biomarkers to work with from that kind of process. So hopefully this is clear. So it's a Bayesian algorithm-based approach looking at causation and inferential causality, linking molecular events to functional readouts, and getting a sense of what to go after when you develop a new molecule or a new drug out there. So through that process, what Berg has done over the past several years now, about 8 years or 9 years now, is we have identified several new targets across various disease areas: metabolic disease, oncology, neurology, vascular biology. Oncology and neurology are our key focus right now. And if we take a target out and we knock down the target or overexpress the target, the chances of getting a functional read is what we call as the success rate. So from that measure, we have about 77% success rate, which means out of some of the 31 targets that we prioritized, 24 targets had a positive validation. And out of those, we have moved 4 of them into drug discovery now. One such program is expected to enter into first in humans in 2020. And then we also have a repositioning program. So now just taking a step back, for the COVID model that we built, this was data that was publicly available from a German lab that published some omics data a month ago. We were able to successfully take the data into our algorithms and then model them with a key focus on identifying causal connections between viral titer, which is a SARS-CoV-2 titer, and the host proteins. And interestingly, one of the key proteins that came out of that analysis, which is again de novo, was ACE2, which actually makes a lot of sense. We have identified at least 10 to 15 other targets that actually make pharmacological sense from what Anton was saying. We are in the process of writing a manuscript for that. So basically, the point is that the approach and the technology that we use is agnostic to any kind of disease or any kind of application that one wants to use it for. And then, in the case of oncology, what we did was this paper that was published in 2019, where a CRISPR study across a bunch of cells was used to identify some of the cancer targets that had a functional relevance or some sort of a functional output to loss of that particular target, either in terms of sensitization or in terms of some sort of a participation in a chemoresistance mechanism. So we use that as our kind of benchmarking for some of the new targets that we discovered through this approach of causal mapping. And so from there, we overlaid some of the Berg networks that we had generated over years from some of the tissue samples that we had acquired with the Behan et al. Networks or the model itself. And we got about 80% match for some of the Berg targets as compared to the CRISPR-Cas9 targets. Of course, there was few that were not overlapping, but most of the targets that were explained or validated by the Behan et al. Study were in the Berg networks as well. So we kind of use that as a foundation for us to kind of triage some targets as we move forward in our process of validation. And so out of that 80% match, we were able to, based on network topology and based on some sort of advanced analysis, we were able to enrich for 47 targets, what we call as Berg priority targets. And the fact that these have a functional consequence to manipulating them, plus there is some clinical data around these targets, we believe that the drugs that are developed against these targets would have a fairly higher chance of clinical success. And so that's our mission, that's our goal, to move some of these targets into validation ourselves outside of the behind a tail study, as well as some of the drug discovery efforts that are ongoing at Berg. Now, so just moving from, just an example of a target, I'm not disclosing the target yet. It's an E2 enzyme in the ubiquitin proteasomal pathway. We call it as BPM42522, that was derived through the same process that I mentioned just a couple of slides ago. Interestingly, the functional correlates of this target, BPM42522, which is an E2 enzyme, is as follows. It is related to microtubule cytoskeletal regulation and reorganization. It's related to nucleosome histone function. It's related to cell cycle regulation and apoptosis and T-cell antigen receptor signaling. The function is extremely, the phenotype of the target, or the function that the target is tied to, is extremely key for us to get a sense of where that particular target could be utilized or could be used as a target. So some of the very high-level data on phenotypic characterization of this particular E2 BPM42522. On the left, we did some siRNA modulation, actually showing that there is an effect on cell viability with the knockdown of these targets in a bunch of cells. And there are a few cells that have no response to knockdown of the target. siRNA, again, confirming on the right corner, confirming that there is an effect of target modulation on apoptosis and cell death. There is also we see a very clear G2 to M cell cycle arrest with a knockdown of the target. Also, when we ran some preliminary studies with human tissue arrays, we actually saw that there's an upregulation of the target expression, especially in the membrane of certain pancreatic tumors, which is significant compared to normal adjacent tumors. So overall, the target has a function, the target has a phenotype, the target is also expressed differentially in human tumors. And so we kind of prioritize this target for the drug discovery. Being a new target, we decided to go with, and again, E2s were considered to be challenging to drug because of the conserved active site. So we went on a discovery mode, trying to identify some novel pockets that we could develop molecules around. We took a fragment-based approach, used commercially available fragment set of 1,500 fragments. Using affinity mass spec and SPR, we were able to come down to two fragments that we could take further into development. Development in the sense, discovery rather, lead ID and lead optimization. Right now, we have finished lead optimization. We have nominated a candidate molecule for development. In the process, what we discovered was that a new allosteric pocket in the protein of BPM42522 that was discovered by this approach. I think the advantage of the RO3 library versus the RO5, where you identify novel mechanisms for drugging. Allosteric mechanisms are not completely new or unknown in the E2 space. There is an example of a E2 homologue called CDC34, where there is a molecule called CC0651 that has been discovered specifically for CDC34, showing some of the on-target effects of this molecule on CDC34. So these targets, I should say, are very amenable to novel allosteric type of regulation. So, I'm just quickly going to skip through the slides. So basically, confirmation of biochemistry on the left, we are able to show that these molecules have effects biochemically. They are selective for a particular E2 called E2 BPM42522. When we ran this across a panel of other E2s, we didn't see much of cross-reactivity. They are strong binders, as you can see on the left corner, whereas they had no binding to the other E2s. So there is some selectivity, specificity. There is also on-target biochemical activity and biophysical activity that we could confirm for some of these leads that we developed. And when we ran them on potency assays in cancer cells, again, we saw a very strong potency of these molecules. In fact, the candidate that we selected is about in the same range as these molecules here. Double-digit nanomolar potency is what we see. This was in Mia-Paca cells, but I'll show you some additional data on additional cells that were used for the potency assays. We don't have effects on normal proliferative cells, like any kind of, in this case, we use the human PBMCs that were unstimulated and stimulated, and we see no effect of these molecules on normal proliferative cells. Again, like in the target, where we actually observed a G2 to M arrest, we actually saw that these molecules were also causing a G2 to M arrest of the target in the cellular system that we used. And as a preliminary read, when we did a sub-Q injection of some of the lead molecule, one of the lead molecules, 206, in a tumor model, this was MDMA231, we actually saw some promising initial results into the tumor growth inhibition activity of these molecules, confirming that these molecules are active. But then after that, with the lead optimization, we were able to get some orally bioavailable molecules that were orally administered, and we have up to 60 to 70% TGI in some of the current studies that we ran. So overall, it looks promising. It's on target, selective, specific. We ran it across kinase screens as well as some of the safety 47 assessments, and it came out pretty clean in terms of what other potential proteins it could be touching. So these are some additional data just to confirm the on-target engagement. As I said, one of the key functions of this protein was a role in cell cycle regulation, and we clearly see that with the molecules. So the molecule recapitulates the effects of the target or the phenotype of the target. We see a G2 to M arrest, as I explained earlier. On the left, I'm showing some data there. In fact, we see some aneuploidy 4N at about 72 hours of treatment, which goes to show the cell cycle effects that the molecule has. We see regulations of cyclin B1, which is a key protein in the G2 to M phase, as well as phosphohistone, which is another important function of the target that I had explained early on. So basically, we see anything that the platform kind of brought us to and predicted in terms of phenotypic outcome of the target, we were able to reconfirm that using the small molecule lead or candidate. And this is a slide I was talking about. So basically, when we ran these molecules across a panel of 102 cell lines, we were able to see robust effect of these molecules across almost 80 to 90% of the cells. I mean, what you see in yellow is the IC50, but if you look at the LD50, when you look at IC50 or the GI50, majority of the cells were impacted, had an effect on proliferation or cell killing. So that goes to show that there is a potential for diverse applications of these molecules for this particular target. We have, ever since then, we have honed in on few disease indications and few disease settings, including one resistance setting where this molecule could have a role. Again, information garnered from the technology of the platform that we kind of use as our foundation. So yeah, so the molecules have good tolerability, they have good DMPK profiles. We're actually starting a discovery toxicology studies on these molecules. Hopefully we'll be able to run IND enabling studies before the end of the year. And so that's pretty much, I think it's my last slide. So yeah, so this is a story about how we use AI, some flavor of AI, namely causal networks, to identify a novel target, took it through the process of validation, drug discovery, development, all the way into IND enabling studies. And there is a whole bunch of biomarkers that will also co-travel with this target and this molecule as we enter into phase 1 and phase 2. A quick note on the fact that this approach, this technology that I just walked you guys through, is applicable to more than just oncology. We just, as I mentioned, we are writing up a paper on COVID-19 and what we learned from that experience. Thank you, and that's my last slide. Thank you, and I'm willing to take questions at this point.
Hello, can you hear me?
Yeah, I can.
Great talk. You mentioned some of your efforts in neurology. Can you expand any upon those?
Yeah, so we have a target that is an oxidative reductase enzyme that we are developing in small molecule for. That is most likely in the Parkinson's disease area. But that's just one example. We are open to we're looking at other potential applications of that particular enzyme, as well as certain other programs. So yeah, our primary goal in neurology is Parkinson's, but we are open to looking at other indications as well.
Thank you.
Hello. Can
Yeah,
you hear me?
I can hear you. Yes.
Okay. Yes, I'm Svetlana Marikan, and I have a question regarding you tested against so many cancer cell lines. Did you try to test against actual primary cancer and see if the effects when you have diverse population, genetic population, how your effects are?
Right. That is a good point, and that is definitely what we're doing right now. Through our collaborations, we are able to access some of those primary cells with certain known mutations, and that I think will kind of allow us to kind of hone in on certain indications that we can, develop the program around.
Okay, thank you.
Thank you. It was a wonderful talk, by the way. And I think in terms of the systems approach, by the way, in that we can take any data. So we are not limited to literature data. I just want to tell you that.
Right, exactly.
So there is a way, where you have to integrate pharmacology, into cause-effect analysis, because when you talk about networks, the background they are working in ultimately determines the network interactions. So in the cause-effect relationships are relative, because you have an adaptive system. So essentially, if you change your background, the cause-effect relationship changes. So cause-effect relationship is a moving target.
Right.
That is one of the issues in a way.
That is very true. In fact, in one of our clinical programs that I've not even talked about, we do take systems pharmacology into account, because we have been collecting samples from a phase 1 trial and phase 2 trial that we have just finished for the clinical program. And one of the things that we pay attention to is, like what you said, systems pharmacology, obviously, because these patients see so many other drugs, there are so many other interactions that happen. And it's actually what that does to the system that we are able to map, rather than like a single slice of a time. So I think I appreciate completely what you're saying. The company also believes in a systems pharmacology approach.